Hello everyone. We'll wait for everyone else to join and let's see. Within a few minutes, we are going to begin. Okay, within 3 minutes or so. Let's see by then how many people join. Okay, so by then I'll introduce myself. Hello, I'm Bias Mukherjee from IIT Kharagpur, and my department is Department of Bioscience and Biotechnology, and my supervisor is Professor Shomo De. And this course, as you know, is coordinated by Professor Sudeep Kumar Ghosh and Professor Agnew Ganguly from Department of Bioscience and Biotechnology, IIT Kharagpur. And this is the very first time for me taking immunology course, okay, at NPTEL. Before this, for 3 years, I have been taking fundamentals of protein chemistry and experimental biochemistry course. So I will also be learning a lot with you people. Okay, you guys will be teaching me, and I will be teaching you all. One second. হ্যাঁ আমার NPTEL ঠিক আছে ঠিক আছে Hello everyone, I'm back. Yes, so I'm back. Hello once again. So I'll introduce myself once again. I'm Bias Mukherjee, a research scholar from Professor Shomode's Lab of Structural Biology and Protein Engineering from the Department of Bioscience and Biotechnology, IIT Kharagpur. And in today's class, we are going to talk about immunology, okay, the basic concepts of immunology. This is the very first time that I'm taking this NPTEL Immunology course coordinated by Professor Ghosh and Professor Ganguly from the Department of Bioscience and Biotechnology. And so if there are any questions that you have, I will share my mail ID with you. You can ask me the questions. If I'm not able to answer in today's class, I will definitely make sure that I answer in the next class, okay? And we are going to go through all of these different topics that the professors have covered in first week. So we are going to brush upon those topics and we are going to solve questions, MCQ questions related to those topics. Okay, so these questions are previous year's questions from NPTEL, this particular course, from GATE and from CSIR NET. And then finally in the revision class, we are going to solve all of these different CSIR NET, GATE, BET questions. Okay, and hopefully this course is going to be very, very valuable to you. Okay, so let's start. Right. So the first and foremost, why do we need to study immunology? Right. So we all are here today, and you know that huge revolutions are happening. You know about COVID. You know about everything in the world that is happening. And why do you think is biopharma and all of these markets such huge? So the basic answer is that we need to study immunology to stay alive in this world. Okay, so immunology is the study of our body and how it protects us against pathogens and different infections. So understanding our body's defense system, understanding our modern medicine, that is vaccine, transplantation, cancer immunotherapy, allergy and autoimmune treatment, all of this is a part of immunology, and it is the central to the understanding of diseases. Okay, so that means this is implicated in cancer, in cardiovascular disease, neurodegenerative diseases, and every other form of infections that go on in our body, and every other form of degeneration, be it aging, be it any other kind of diseases. Okay, and most importantly, this is important for you all, that the this drives biomedical innovation. So there is a lot of career prospect in this field. Okay, so biologics and immunotherapies are important and they are the fast growing drug classes. So there's a lot of career prospects in immunology and related fields. Okay, so we will move on to the history of immunology. Now this professors have already explained, you guys also know possibly, so we will go through it very, very quickly. Okay, so in the ancient times there was something called variolation. Now variolation is it's a very gruesome procedure, if I will be honest with you. It means that the pustules that were formed in cowpox and in smallpox, so healthy people's skin was rubbed against these diseased pustules, and in that way this immunity was transferred to this healthy person, and that is what was practiced in China and Middle East. Very crude way of uh, immunization, but yeah, that is how it was. Okay, then Edward Jenner, he demonstrated that instead of smallpox, now smallpox happened in humans. So instead of smallpox, if you take cowpox pustules and all, and if you introduce that into healthy human beings' body, then also it is helping to cure their smallpox. Okay, so that, and then he gave the name vaccine. He is known as the father of immunology. And then in the late 1800s, Louis Pasteur came. We all know about him. He was the father of microbiology, right? And then Robert Koch came up with your microscope. And then what happened, a lot of different people started coming up with a lot of different studies. So we came to know about phagocytosis, we came to know about antitoxins, okay, and humoral and cellular immunity, and all of these different topics came to be known gradually. And then in the 20th century, the complement system was identified, and T cells, B cells, this was discovered. MHC, that is major histocompatibility complex, was studied for the first time, and molecular immunology came into the picture. Okay. So now we will move on to understanding immunology. Now, immunology is the study of the immune system, the tissues, cells, molecules involved in innate and adaptive immunity, and the host defense systems that is there to protect us against all kinds of infection and the adverse effects of these um, defenses. Okay. Now, the golden rule of immunity is that we want to kill the pathogen, but we do not want to harm the host. Okay. That is what we have to keep in mind as we study all of these different immune cells. The basic purpose is that. But a lot of times you know that pathogens are there inside the immune cell, right? For example, there can be these Mycobacterium tuberculosis which can be residing inside your macrophage. Then what will you do? You will have to kill the bodily cell to kill that particular organism, right? It is not going to come out of that cell. It's a virus. So that is how it is going to be there latent for years and years and years. And there we have no other option but to kill the host. Now, two core jobs of the immune system: protect the body against immune diseases, that is virus, bacteria, fungi, parasites, and distinguish self from non-self, that is my own body cells from other pathogens from outside, right? And eliminate potentially destructive foreign substances. Now we go to innate and adaptive immunity. This also we have studied since school. So innate immunity is what? It is there inside our body, and this is very generic. It helps to protect from everything. But adaptive immunity is a lot more specialized. This takes a lot of time to develop, but then it is very, very specialized and very, very efficient. So all the B cell, T cell, etc., this comes under this category. And what happens is, this is very useful in eliminating the foreign pathogens and invaders very effectively. Okay, so speed of response is definitely very fast for innate immunity and very much delayed for the adaptive immunity. Specificity, I told you, this is generic and this is very specific. Then you have memory. Now innate immunity does not have memory, but then you know about memory B and T cells. So these store the memory of the pathogen, and then when there is a second time infection, very, very rapidly the antibodies start getting produced, all the immune cells, they start getting activated, and instantly the infection is removed, right? So the pathogen is killed. And the key cells, so which all are involved. Now macrophages, neutrophils, dendritic cells, NK cells, all of these are involved. Coming to adaptive immunity, majorly B cell and T cell are involved. Now, another very interesting point is that adaptive immunity and your innate immunity, these are not very distinct entities, okay, because there is a constant interplay going on between these. Okay, so there is a constant crosstalk going on, and initially it starts with innate and it gradually moves on to the adaptive immune response. Okay, now recognition tools, you know that there are these pattern recognition receptors. For example, there are DAMPs and there are PAMPs. So we will be talking about all of those. So our body cells recognize using these pattern recognition receptors that there is certain pathogen in the environment. And coming to the adaptive immunity, now this B cell and T cell, they have specific receptors. They have their co-receptors. All of these very, very well established system is there, and it is armed to attack the pathogen. Also called first line of defense, and this is specific or acquired immunity. Okay, so both work together, and that is how we are still living after millions and billions of years. Okay, so we'll start with the innate immunity. So if you see that innate immunity has 4 different type of barriers. The first one is called anatomic barriers. Okay, so this table has been taken from QB Immunology book. Please refer to QB. It's a book where everything is explained beautifully, but then I understand, I was also a student like you once, and I understand that it is very tiresome to go through a book initially. So that is why I've made the slides in such a way that whatever questions are there in this slide, if you read the slides properly, read the presentation, you'll be able to answer those questions. Okay, but for in-depth knowledge, you'll have to refer to the book. All right, so we'll go on. Now, so skin, you know, this is the biggest, the largest barrier, and this is very effective also. And something very interesting I'll tell you, that on our body there is this bacteria called Staphylococcus epidermidis, and this bacteria is a, it is naturally present in our body, and this prevents this Staphylococcus aureus, which causes pimples on our face. This causes all these infections, right? So this epidermidis prevents this S. aureus to come and infect us. So that is how the skin is a protective barrier, plus all the microbes in the skin, they are also protecting us from harmful microbes coming from outside. Okay, so that is how everything is a very beautiful balance. Now see what is there. So there is this acidic environment that is there in our stomach, right? You know, the pH of HCl in our stomach is 2, and that is where all the food microbes that we are taking inside through food, or the microbes that come in through food, they are getting killed there. Okay, and then there is a mucous membrane, and it has lysozyme also. It is killing the bacteria. Tears are having lysozyme, right? And then the physiological barrier, so there is temperature, the body temperature is relatively higher, you know, and then when we have fever, the temperature is deliberately increased and the microbes are killed there. Low pH, stomach pH, chemical mediators, lysozyme, that is there in our tears, in our saliva. Okay, then there are interferons, there is this complement lysis of microorganisms. We will be talking about all of this eventually. Okay, and then there are these toll-like receptors which recognize, these are the pattern recognition receptors that recognize the pathogens. Then we come to phagocytotic barriers. Now what are phagocytocytes? Now phagocytotic cells are macrophages, dendritic cells. They all come and they engulf microorganism and eat them up. Okay, phagos means to eat. They come, swallow, eat up, right? Then there is the inflammatory barriers. Now inflammatory barriers, here all the other cells are involved. Your neutrophils are largely involved, and then a lot of chemokine and cytokine, everything is involved, so other cells are called upon. And then this is the point where adaptive immune system is also started, so its response is also started. Okay, we will read about all of these in details. Okay, now origin of the immune cells. Now we know that there are so many immune cells which are so difficult to remember. Now from where are they emerging? So all of them are coming from human bone marrow. So inside the bones, all of these cells are starting off as hematopoietic stem cells, and these cells are dividing into the common lymphoid progenitor and the common myeloid progenitor, and the lymphoid group, this gives rise to B cell, T cell, all your adaptive immune cells, and the myeloid progenitor, this gives rise to the innate immune cells. Okay, immature dendritic cells, there are these, again these cells divide once again to give rise to monocytes, macrophages, eosinophils, basophils, right? All the red blood cells, white blood cells, everything that you have studied about. Okay, so we'll go on to the next slide, and there we will see how these immune cells are differentiating. Okay, now we'll go to the next part. Right, so we have 2 MCQs, so that you people don't get bored, don't sleep off, and don't run away. I have these MCQs after a few topics. Okay, so try to answer in the chat box. The first MCQ, so you can answer as 1 and 2. Okay, so the first MCQ tells, which of the following best describes the defining features that distinguish adaptive immune system from innate immune system? Presence of phagocytotic cell, immunological memory and antigen specificity, involvement of the complement system, and ability to cause inflammation. Okay, and the second question is, all cellular components of the blood, including immune cells, arise from which precursor cell in the bone marrow? 1B, 2B. Okay, very good. Let's look at the right answer. Yes, very good. So you are paying attention. You're not sleeping. Congratulations. We move on to the next slide. Yes, very good. Stay active. Answer the questions. You will not feel sleepy. Chalo. So principles of the innate immune system. So some microbial populations in our body. For example, gut microbes, right? So now gut microbiota, a lot of research is going on regarding this. Now it is even said that your gut microbiome is controlling your brain, it is controlling how much you are able to concentrate, it is controlling how your mood is changing with respect to whatever you're eating, right? So gut microbiota is a huge topic of research now. Okay, and skin flora. Skin flora, I told you already about Staphylococcus epidermidis, which is naturally present in on our skin, and Staphylococcus aureus, which is a pathogen. Right, so these do not cause host damage. They are majorly commensals, that is, they are not causing any damage to our body, or they are in fact symbiotic also. Because a lot of this gut microbiome, they are releasing a lot of compounds by digesting the food that we eat, and these compounds are important for our own body. Okay, they are important for the digestion of food and a lot of other different things. Okay, now anatomic and chemical barriers, these are the first line of defense against pathogen. Innate immunity is triggered by inflammatory inducers signaling pathogens or tissue damage. Now the myeloid linkage makes up the innate immune cells that I told you, and the sensor cells. The sensor cells are the pattern recognition receptors that are the most important people to distinguish between self and non-self. Okay, so sensor cells trigger inflammation by releasing mediators such as chemokines and cytokines. Now what are these? Now chemokines and cytokines, these are chemicals and these are different kinds of peptides and all, and these are actually calling other cells and other things to come. Okay, now NK cells and innate lymphoid cells. NK means natural killer cells. Okay, so they are effector cells that are related to adaptive lymphoid lineage. So adaptive immune response. Okay, now we will look at phagocytosis. Now phagocytosis is a very straightforward thing. What happens is initially a bacterium comes and sits. As soon as it is recognized, the macrophage that is there, its outer membrane, it goes and completely surrounds this bacterium, engulfs it and forms a phagosome. This is kind of a vesicle and it is a phagosome, and then this phagosome goes and fuses with the lysosome. Lysosome is the suicide bag inside the cell, this you know. Okay, and it releases a lot of lysozyme, a lot of proteolytic enzymes, and this forms a phagolysosome which completely degrades this particular bacteria. It is gone, it is dead. Then what happens, all the degraded bits, they are taken and thrown out of the cell. These can be even presented on the top of the cell, showing that there was this particular thing which had infected and I have eaten it up. So using this MHC, which is a major histocompatibility complex, it is a kind of receptor on which these processed antigens can be stored. So we will talk about that later. Okay. For now, understand this: monocytes, neutrophils, tissue macrophages perform phagocytosis. Phagocytose pathogens, for example, microbes, dead cells, and clean the mess. Okay, so this is very important in our body. Now, macrophage can also act as antigen presenting cell. So, antigen presenting cell means, okay, what is so funny? Okay, so antigen presenting cell means that it has destroyed this microbe and now it is taking all the antigens from the microbe. So small pieces of proteins that are there in the microbe or anything, it is presenting it on the surface. That is an antigen presenting cell. Now this is very important because this is going to tell the other immune cells that look, this had come inside our body, I killed it, you be aware that this can attack again. And if there are a lot of macrophages that are displaying a lot of these same antigen, then the body understands that no, this is a time to be aware, and the B cell, T cell maturation begins, that we are under attack, we have to be careful now. Okay, that is the full process. Okay, now what happens? Let's say that there is a wound. Okay, you were walking by somewhere, some tree branch was there, poked into your hand, and that is what we can see, tissue damage. So what happens when there is a tissue damage? A lot of, you know about inflammation, of course inflammation happens. So what are the four different things properties of inflammation. The first one is rubor. Rubor means redness. Then there is calor. Calor means heat. So the area gets heated up. Why does it get heated up? Because there is a lot of blood flow happening towards that point. That is why it gets heated up. Then there is dolor. So dolor is pain. Now, how will you remember this? You can see it this way, that no pain, no gain, and dollar is money. So for money, you'll have to work hard. So dollar and pain. And then there is tumor. So tumor means swelling, right? So these are the four properties of inflammation. So what happens in inflammation? Let's see. So tissue damage causes release of vasoactive and chemotactic factors. What is a chemotactic factor? Chemotactic means it is calling other cells to come to you, okay? And it's a chemical that is telling other cells to come to you. Vasoactive means it is acting on the blood vessel, right? And trigger a local increase in blood flow and capillary permeability. So it is broadening the blood vessel. Permeable capillaries allow influx of fluid and exudate. So what is exudation? Exudation means fluid is oozing out from the blood vessel and going to the site of injury. Okay, and if you see this yellow thing is actually your neutrophil. So neutrophils are most active when it comes to innate immunity, inflammatory barrier, and they are going and they are extravasation is happening, that is through the epithelial lining. It is squeezing between the epithelial lining, going through diapedesis, that is the process of going to the site of infection. Okay, and the phagocytes migrate to the site of inflammation through chemotaxis, and the phagocyte and antibacterial exudate destroy the bacteria. Then what happens? Then the entire blood clotting activity, entire thing happens. Okay, a lot of different cells are called to this place, and they are stopping the blood from oozing out, they are killing the bacteria, and a lot of different things are happening. So we will talk about this in details. So see, inflammation begins when the tissue resident macrophages and mast cells detect PAMPs and DAMPs. What are PAMPs? PAMP is pattern. We have this full form in the upcoming slide. Okay, so we will talk about, we will read it there. So PAMP is more or less pattern something. Okay, you'll see very soon. Okay, via TLRs. TLRs are the toll-like receptors which recognize these PAMPs or patterns that are there in the microbes. And then disease associated, again MP you will see. Okay, then what happens? A lot of histamines, prostaglandins, cytokines, all of these are released. Then what happens? Complement anaphylatoxin. Complement system we will be studying in the upcoming classes. Okay, so right now I will not be explaining complement system, but this is something very, very important, and this is a huge topic, very interesting, and very, you'll remember it. You'll have to remember it. Okay, now these mediators cause vasodilation, that is dilating the blood vessel. Redness and heat is caused because of this. Increased vascular permeability, allowing the plasma and the proteins to leak out as exudate, resulting in swelling. Then there is bradykinin and prostaglandins which sensitize the nerve endings, causing pain. That is why dolor. Okay, as the blood vessel slows, that is stasis happens, leukocytes, white blood cells, they are migrating towards the vessel walls and undergo extravasation via adhesion cascade, that is rolling, or selectin. So selectin is also a kind of protein. Okay, firm adhesion, selectins and integrins, these are basically proteins that are there on the membrane. And diapedesis means transmigration through the endothelium that we saw. Okay, and then what happens? It goes to the site of the injury. Neutrophils arise first, followed by monocytes which differentiate into macrophages, and then a lot of different things happen that you can see here. So neutrophil, extracellular traps and all that, all of this is happening. Then something called opsonin comes. Now this is very interesting. What is opsonization? So for example, let's say this is the bacteria which got in. What is opsonization? A lot of antibody will come and stick to this bacteria, and it will completely coat this entire bacteria. And what happens? This entire bacteria is coated, and then the entire bacteria is killed. It is either phagocytosed by the macrophage and it is killed. So that is how opsonization is happening. This process is called opsonization, right? So now what happens? So then what happens? Neutrophils undergo apoptosis, anti-inflammatory mediators are all there. So you can understand from here that a huge cascade of events have happened. Okay, so inflammation is something that is very important for the body because this is the first sign that something is going wrong. Okay, now all the 6 stages of inflammation are explained here. All the key players are there, what happens and the result. So you can go through this uh, thing whenever you get time. Okay, whenever you get time, go through this table. Now we come to the anatomic barriers, complement and host strategies. So there are 3 different ways the microbes can be dealt with. The first one is avoidance, prevent entry altogether; resistance, that is block or destroy the microbe; and the third one is tolerance, that is allow limited growth without harm. Then we have the first line of defense, which is the skin, and then oral mucosa, respiratory epithelium, all of these, right? Then you have the complement system. Complement system is made up of a group of proteins, a huge group of proteins which are heat labile. They are non-specific, so they are a part of the innate immune response. They directly kill bacteria through opsonization, and also complement system itself is capable of forming a channel. So let's say this is the bacteria, it will form a channel on the surface of this bacteria and it will get inside and it will kill it. Okay, so that is how it can rupture the bacteria basically. It also assists antibody coated pathogens. Complement activation discussed later, we will discuss about this. Okay, now phagocytosis and inflammatory barriers. Phagocytosis we saw, and inflammatory barrier also you saw. Now granulocytes include all the WBCs that are having granules in them. For example, eosinophil, basophil, etc. Right? NK cells kill virus infected cells. Remember this. Macrophage, dendritic cells, neutrophil recognize and phagocytose foreign pathogens. Inflammatory inducers are bacterial lipopolysaccharides, ATP and urate crystals. So these induce inflammation. Okay. Now we will talk about the innate immune cells. Macrophage, you know, important for phagocytosis. Dendritic cell, once again, important for macropinocytosis. What is this? Pinocytosis is drinking fluid. So I told you that phagocytosis is engulfing, right? Pinocytosis or pinocytosis, it is drinking up fluid. And macropinocytosis is when the entire antigen is engulfed. Okay, and then what happens is neutrophil. So neutrophil, so this captures antigen at peripheral sites through macropinocytosis and migrates to the lymph node to present the antigen to the B and T cells. Okay, then you have neutrophil. So what is neutrophil doing? Neutrophil is a fast acting phagocyte which it is the primary killer at the site of infection, mainly works on virus infected cells. Okay, so all the 3 are antigen presenting cells, but the dendritic cells are most efficient at the adaptive immunity uh, introduction. Okay, now eosinophil, basophil and mast cells. Eosinophil, remember, it is very important for killing large antibody coated parasites, especially helminths. Okay, releasing toxic granular content. The granules they are made up of toxic contents. Now basophil circulates in the blood, promotes allergic responses and augments anti-parasitic immunity. Mast cell, remember this, it is also having a lot of granules with histamine. Now histamine, you know that this is linked with allergic responses. Okay. So together with macrophages and neutrophils, the granulocytes form the myeloid arm of the innate immune system, and they are specialized for a different class of threat. Okay. So now we will talk about the PRRs and TLRs. Okay. So these pattern recognition receptors, they can be pathogen associated molecular pattern. I forgot the name. You don't forget, please. Okay. So that is PAMP. And what is DAMP? DAMP is damage associated molecular pattern, or DAMP. So this tells you about the pathogen that has entered. Now macrophages and dendritic cells carry surface receptors that bind to the microbial molecules absent from our own cells. That is how they recognize between the self and non-self cells, right? Now, mannose and glucan receptors. So, bind to the cell wall carbohydrate of bacteria and yeast and fungi. Then you have the TLRs. TLRs are a key pattern recognition receptor family on macrophages. So these are present on macrophages, dendritic cells and other immune cells. TLR1 and 2 heterodimer binds to lipopeptides from gram-positive bacteria, and TLR4 binds to lipopolysaccharide from gram-negative bacteria and lipoteichoic acid from gram-positive bacteria. Okay. Now, remember the names and try to answer these two questions. Okay. First question, please answer as 1 and 2 once again, because I don't expect you to remember the number. I myself don't remember. Okay. So for the first question, it is toll-like receptor TLR4 primarily recognizes which PAMP? TLR4. And the second question is a single antibody molecule IgG can bind to maximum of how many antigen molecules? If you remember, so we are going to talk about this in the upcoming slides, by the way. If you remember, IgG is a monomer, right? IgM is a pentamer. IgA is a dimer, right? That is how you're going to remember everything. So if it is a monomer, it is having 2 FAB regions, then how many antigens is it going to bind, or how many, let's say, epitopes it is going to bind? Okay, try to answer. 1C and 2B. Okay, let's see. Yes, you're paying attention. Very good, I'm glad. Okay, very nice. Now we move on to inflammation. So the inflammation is the body's first alarm system. So what happens is macrophage recognizes and eats invading bacteria via its receptors. What are the receptors? Toll-like receptors, right? Now, activated macrophage releases cytokines and chemokines. You can see that in this figure. These mediators vasodilate the nearby blood vessels. Neutrophils and other immune cells are attracted, and more immune cells accumulate at the infection site, and inflammation is a part of the innate immune response, but this also initiates the adaptive immune response, and that is why it is so important. Okay. Now we will move on to NK cells and ILC. So NK cells, you know, this contain lytic granules that are containing a lot of perforins. So what is perforin? Perforin makes holes inside the cells or inside the... Now this one actually kills virus infected cells, remember. So let's say this is the cell, it is infected with a virus. This is my cell, okay, it's infected with a virus. NK cell comes and it recognizes it and it releases these perforins, and what happens is the cell is killed. So kill virus infected cells and tumor cells behave like cytotoxic T cells but lacks antigen specific receptor. So that is the only difference between a cytotoxic T lymphocyte and a natural killer cell. Otherwise both of them kill virus infected cells. Okay. Now innate lymphoid cells, these mirror the functions of Th1, Th2, Th17 helper cells. Now T cells, we will talk about this in details in the upcoming slides. Okay. Now T cells can be helper T cells. T cells can be cytotoxic T lymphocytes, right? Two different types. Now helper T cells also, there are different types of helper T cells. So these ILCs basically are similar to them. Now, they reside in the lymph nodes and tissues, sensing infection and responding rapidly. They bridge the functional gap between innate and adaptive lymphocytes. Okay. Now, we move on to adaptive immunity. Adaptive immunity is a lot more complex, so please pay more attention now. Okay. Now, antigen binding to an antigen receptor. The B cell and T cell, they have receptor called B cell receptor and T cell receptor. Okay. Drives the lymphocyte to become effector and memory cells. So they basically mature and they distinguish. So they basically divide into effector T cell or B cell and memory B cell and T cell. Now antibodies and TCRs both have constant regions, FC region, you know, and the effector function, and variable region, that is the antigen binding region. Now, BCRs and TCRs recognize antigen by fundamentally different mechanisms. So, receptor genes are built by somatic gene rearrangement of incomplete gene segments, generating enormous diversity from a limited gene pool of very few genes versus millions of possible different antigens. So now there are so many antigens in the world, but then we have very limited genes. So how is it that we produce these antibodies that are specific to all of these different antigens that are there in nature? How is that even possible? So the answer is, we will talk about that, and the answer is somatic gene rearrangement of incomplete gene segments. So we will talk about this in details in upcoming lectures. Okay. Now, antigen activated lymphocytes proliferate into clones of antigen specific effector cell. Now the effector cell that is getting formed, this is containing the receptor that is specific for particular antigen, that specific antigen. Okay. Now, self-reactive lymphocytes. Now, this is very important. This is normally deleted or functionally inactivated during development. Why? Otherwise, autoimmunity will be triggered and the body cells will be killed off. Right? That is why it is very important to distinguish and kill them if they are reactive towards the body cells and body proteins, antigens. Now, antigen, antibody, epitope, paratope. Very straightforward things, please don't confuse. Okay. Antigen, you know, this is the foreign molecule. Antibody is the Y-shaped thing that binds to this foreign molecule. It is produced by our body. So the protein produced by B cells or plasma cells, right? Epitope. Epitope is the region in this antigen that binds to the antibody. The specific part of the antigen that is recognized by the antibody. Paratope, the antigen binding site on the antibody. So epitope and paratope actually interacts, and in the bigger picture it is the antibody and antigen that is interacting. Why so many antibodies? Now millions of possible antigens exist, and somatic gene rearrangement is the way we get complementary binding antibodies for all of these different antigens. Now the B cell receptor and T cell receptor. Now B cell receptor is a perfect Y. Okay. So the Y-shaped two identical antigen binding sites, it is bivalent. Bivalent means that it is going to bind to two epitopes. Correct? That is why bivalent. Binds native or unprocessed antigens directly. What is unprocessed? Unprocessed means that, you remember, antigen presenting cell, it ate up, then it degraded, then it presented on the surface. That is called processed. Unprocessed means directly in nature, as soon as it enters into the cell, it is eaten up. And that is what this is. So it is not eaten up, it is basically binding to this. So B cell receptor can go and bind to the antigen, native antigen. Okay. When this B cell receptor is secreted from the B cell develops into a plasma cell which gives out 2000 antibodies per second, and these antibodies are also exactly same as the B cell receptor, only the transmembrane region is missing. That is why it is not bound to it, but it is released. Okay. Hinge region allows flexible movement. Now you come to T cell receptor. T cell receptor has two chains. One is called alpha chain, one is called beta chain. It has only one antigen binding site. Cannot bind whole or native antigen. So it only binds to processed antigen. Only processed antigen. Needs processed peptide, that is chopped antigen presented on MHC. Always membrane bound, never secreted, and the chains are linked by disulfide linkage. You can see that in the figure, right? Okay. Answer this MCQ. Two MCQs are there. Okay. The first MCQ is, which statement correctly differentiates recognition by B cell receptor or BCR from the T cell receptor or TCR? Both recognize antigen only processing and presentation by MHC. BCR recognizes native or unprocessed antigens directly. TCR recognizes processed. Yeah, okay. So 1 is B and 2 is C. So 2 says that the part of the antigen that is specifically recognized by, yes, very good. So you guys are actually listening. Okay, great. This is the right answer. Okay, that you can see, and explanation is also given to you in case you are not able to catch up Okay. Now antigen processing and MHC presentation. Why T cells need MHC? Now we said that it needs, but then why? So T cells cannot recognize a whole native antigen the way B cell does, and the antigen must first be processed, that is chopped into a small peptide and then presented on the MHC, and T cell receptor recognizes this MHC plus peptide bound together and not the peptide alone. So what is this MHC? Now MHC, we will talk about this in the upcoming lectures, but for now understand that MHC is major histocompatibility complex. Okay, this has, so MHC has lot of different classes. We'll talk about two. One is MHC1 and MHC2. MHC1 has one transmembrane domain. MHC2 has two transmembrane domains. MHC1 presents intracellular or endogenous proteins. For example, the virus infects the cell, virus releases its own proteins, and this is now considering this is cellular protein only, because our cell is only producing this, right? The virus has hijacked our cellular machinery. So this is presented in the form of intracellular or endogenous protein, MHC1. And MHC2, two transmembrane domains are there, and this presents extracellular or phagocytosed antigen. Got it? Okay. Now what happens is, now this major histocompatibility complex is a set of cell surface protein that display peptide fragments or proteins of self or non-self to T cells for immune surveillance. Okay. Now B cell antigen presentation. So B cell recognizes native unprocessed antigen via their BCR. No MHC is required, but T cell, remember, cytotoxic T lymphocyte, which is also called CD8. So CD8 is a co-receptor that is there besides this T cell receptor, and this together helps in identification of MHC1 presented peptide. Okay, and the helper T cells, that is having CD4 co-receptor along with the T cell receptor, this recognizes MHC2 presented peptide. Okay, so if you look at this, you will see that there is a vast difference. Once again, all the images have been taken from QB Immunology. Please refer to that. Okay. Okay. So see, antigen enters from outside. Then which one will it be? MHC2, right? So it enters from outside. It has been degraded. MHC2 is presenting it, and virus has entered, virus has lived inside us, virus has produced viral protein, everything. Viral protein is going, getting packaged, it is going in the form of vesicles, and then what happens? MHC class 1 identifies it, and MHC class 1 from the endoplasmic reticulum, it is sending it via Golgi, via vesicle, and to the surface of the cell, and there it is getting presented in the form of peptide by MHC class 1. Okay, this is going to be identified by cytotoxic T lymphocyte. Okay, now we are going to talk about primary and secondary lymphoid organs. Okay, so primary lymphoid organ is the training ground. So the bone marrow is where all the cells are first born. Okay, so this is the place where hematopoietic stem cell is there. Now what happens? B cells develop in the bone marrow, but T cells go to thymus. Where is thymus? Thymus is in your chest, okay, behind the heart. So it goes to the thymus. There it starts maturation, and then after maturation, once again it gets into the blood, circulates. Now self-reactive lymphocytes are eliminated. Remember that. Secondary lymphoid organs are the operating bases. So lymph nodes, spleen, MALT and GALT. What is MALT and GALT? Mucosa and gut associated lymphoid tissue. These are the regions where a lot of these immune cells are there. Okay. Mature lymphocytes reside here and encounter antigen. Adaptive immune responses are initiated here by antigen presenting cell. Okay. What is a mature immune cell? Mature immune cell is a one that has been cleared by clonal selection. So it is not going to recognize the self antigen, self peptides. That is a mature one. So we will talk about this in a lot of details. There is a lot of genetic recombination and everything that will happen, okay, in the upcoming lectures. For now, understand this. Okay, so antigen processing and MHC presentation. Why T cells need MHC? So T cells cannot recognize and all this we already discussed. And MHC class 1, okay, this also we have discussed. So here you can see that this is how the presentation happens, and this is how the T cell comes and recognizes using its T cell receptor. And this another tiny thing that you're seeing, this is the co-receptor, that is CD8 co-receptor, and this is your CD4 co-receptor, right? So that is how they are recognizing these presented cells. Then we go to lymph node. What is a lymph node? Now lymph node is a place where there is an afferent vesicle and there is an efferent vesicle, okay, and then you have antigens enter via the afferent lymphatic vesicle. They are carried by the dendritic cell. So what happens is when there is this wound, dendritic cell goes and recognizes. Then dendritic cell engulfs it, presents the antigen on its surface, comes to the lymph node, right? It comes to the lymph node to make everybody aware that this has happened with me. Now what happens? Through the afferent lymphatic vessel, it comes specific T cells that are there in the cortex. So cortex is having a lot of T cell, germinal center is having a lot of B cell. Okay, what happens is the specific T cells recognize and bind the antigen and proliferate. And what happens? Activated T cell help the B cell. So B cells again proliferate in the germinal center and become plasma cells. So plasma cells secrete antibody which drains out via the efferent vesicle this time. Right, so it is coming in through afferent, going out through efferent. An analogy, see, lymph nodes are like local police station, which is what sir has explained in your presentations, right? So that is how sir had explained, and handling the infection in their own jurisdiction of the body. Okay, now we go to the secondary lymphoid tissue, that is spleen. So what happens from the lymph node? We have understood what happens in the lymph node. Now what happens finally is that in the spleen, it filters all these blood-borne antigens and has T cell and B cell zones plus germinal center like in the lymph node. MALT and GALT, this covers the mucosal region with no direct blood or lymph node access, and it induces tonsils. So these are also regions where a lot of your T cell and B cell are present, okay. So tonsils, adenoids, appendix, Peyer's patches, NALT. NALT is nasal associated lymphoid tissue. BALT is bronchial associated lymphoid tissue in the lungs, okay. Specialized M cells. M cells are there in the Peyer's patches, and we will talk about this later once again in a lot of details in the upcoming classes, okay. Right now, we'll just brush upon all these topics, okay. And tonsils and etc., they protect the mouth and throat. That is why they swell during a throat infection, right? Inflammation happens there. Now, okay, you have 2 MCQs here. Try to answer. Which secondary lymphoid organ is primarily responsible for mounting immune responses against blood-borne antigens? Thymus, bone marrow, spleen, and Peyer's patches. Then we have, okay, one is C. Okay, good. And then what about the next one? In a lymph node, the germinal center is primarily the site of T cell maturation. Okay, C. Okay, let's see. Let's see which is the right answer. Yeah, so your right answer is spleen, and then B cell proliferation and antibody affinity maturation. Okay. All right. Moving on. Now we come to something that is important. You will have to understand this. This is humoral and cell-mediated immunity. Okay. In humoral immunity, what happens? Okay, we'll start with cell-mediated. So see, internalized antigen digested by cell. Okay. Then what happens? Altered self-cell presents antigen. Antigen is basically presented, right? Then what happens? The T cell receptor comes, recognizes the antigen bound to the MHC molecule. Binding antigen MHC activates the T cell. So T cell basically gets activated. Then what happens? The activated Th cell secretes cytokines that contribute to the activation of B cell, T cell, and other cell. Okay. Then what happens? Acha. So if this is Th cell, that means what MHC was that? Try to recall. If it was TH cell, then it's there in the figure. You can see. Chalo, answer. It is class 2 MHC if it is TH cell. Okay, now see what happens. So activated TH cell activates all other cells. Okay, then what happens? Activated cytotoxic T lymphocytes recognizes and kills altered self cells, and these are the ones that are presented by class 1 MHC, because CTL will only bind to class 1 MHC presented peptides. Okay, now all these cytokines and everything were released. Then what happens? The B cells interact with the antigen and differentiate into antibody secreting plasma cell. B cell directly interacts with antigen. It does not need presented antigen on the surface, correct? Then what happens? These plasma cells, they are releasing all these antibodies. The antibodies go and coat all the pathogens and kill them by opsonization. So antibody binds antigen and facilitates its clearance from the body. So that is your cell mediated and humoral immune system. Okay, now dendritic cell, the bridge to adaptive immunity. Dendritic cell, immature dendritic cell reside in peripheral tissue, for example skin. On infection, they capture antigen by micropinocytosis, forming a macropinosome. They migrate to the nearest lymph node, maturing along the way. In the lymph node, they present the antigen via MHC to the surface of the T cell. Specific T cell binds, get activated, proliferate, and helps B cell to get activated. Okay. Now, three professional APCs are dendritic cell, macrophages, and B lymphocytes. Okay, so I hope the story is clear until now. So we will talk about the T effector cells quickly. Okay, now cytotoxic T lymphocytes or CD8 co-receptor they are having. They recognize MHC1 peptide presented, which is through intracellular or endogenous proteins that are presented on the surface. Okay, target virus infected cell and tumor cells expressing abnormal proteins. Now remember, virus infected cells and tumor cells, they will be presenting abnormal proteins on their body. That is what it identifies. Okay, now deliver a death signal, that is perforin. I told you about perforin. This is a pore forming complex, pore forming chemical, right? Then there is granzyme. Granzyme triggers apoptosis. And then we have the Th1 response, which activates macrophages. Some bacteria, for example, Mycobacterium tuberculosis and Mycobacterium leprae, survive inside the macrophage by blocking the phagosome-lysosome fusion. So bacteria is evolving much faster than we are evolving, right? That is why bacteria is able to, that is why you have this antimicrobial resistance in our body. So bacteria is basically getting resistant to all the drugs that are giving to kill it, and that is why we are kind of helpless right now, right? So bacteria is staying inside the macrophage. Bacteria is producing its own proteins. So Th1 cell recognizes the bacterial peptide on MHC2. So bacteria's own proteins and the macrophages changed proteins, all of these are getting presented on the MHC2 on the surface of the macrophage, and then Th1 release interferon gamma. Interferon gamma signals the macrophage to fuse its lysosome with its phagosome, destroying the intracellular bacteria. Okay, that is the work of Th1 cells. Coming to helper T cells, they recognize MHC2 peptide and from the extracellular antigen that has entered into the body. Right, phagocytosis or extracellular antigen, it is having CD4 co-receptor. Now another one is helper T cell or follicular T cell. This is the one that helps to activate the B cells. Okay, so B cell needs a second signal from the helper T cell via MHC2 to get fully activated and become plasma cells. Now everything that we talked about is explained in this diagram, right? So if you look at these diagrams properly, you will be able to understand. Okay, right. Now there are two MCQs for you. Which of the following correctly pairs the MHC class with its associated T cell co-receptor and typical antigen source? Okay. And the next question is, natural killer cells differ from cytotoxic T cells in that NK cells. What is the role of NK cells here? And how is it that NK cells differ from cytotoxic T lymphocytes? We discussed this in today's class. Try to recall. One is C. Okay. So T cell co-receptor and okay. What about the next one? Requires MHC class 1 presentation of antigen to function, expresses antigen specific receptors by gene rearrangement, do not require prior antigen specific activation and lack rearranged antigen receptor. Okay. Okay, let's see. A lot of you are answering. That is very good. Okay, we will look at the right answers. So for the first one, yes, you're correct. It is C. So why is it C? Because MHC class 1 molecule has one transmembrane domain. It is presenting your endogenous cytosolic protein, that is viral infected cell or tumor proteins synthesized inside the cell that are recognized by the CD8 plus cytotoxic T cells. And MHC class 2 molecules have two transmembrane domain and they present phagocytosed antigen or exogenous antigen, and they are recognized by the helper T cells. Okay. Now, natural killer cells, how are they different? They do not have this T cell receptor, right? That is the difference. They do not require prior antigen specific activation and lack a rearranged antigen receptor. Okay. That is the story. Now, okay. So now what happens is, we'll talk about vaccination. So what happens is, initially when we all were kids, you remember it was a very painful procedure where our parents used to take us to the doctor and they used to inject. There were so many injections in your childhood, right? Every month something or the other, and it was so painful. And then the child used to have fever, the child used to cry, they were in so much pain. And what used to happen was, it used to take a lot of time to for this pain to reduce and everything. But then that was necessary. Why? Because we take these vaccines, and that is our primary exposure, right? So when we take the vaccine, there is this attenuated microbe or killed microbe, and it is our first exposure. So at the first exposure, there is a lag phase. Now, if you remember bacterial culture, you will know that when we inoculate bacteria into a media, it initially takes a lot of time to get adapted to the media, and then it starts growing, it goes into its log phase. So initially this is a lag phase and then it is the log phase, right? So it is the same for this microbe, because this is also after all a bacteria or virus or anything, right? So what happens is, initially there is a lag phase. So antibodies take around 7 days to form. Okay. And so it is very, very stable way it is growing. Then what happens when there is a secondary infection, that is when this child later on grows up and the child actually catches this particular pathogen from the environment, let's say it is chicken pox or anything. Okay. Then what happens inside the body, the antigens are, the antibody is already prepared, plasma cells are already prepared. So as soon as the antigen is detected, instantly the body starts producing all of these antibodies, plasma cells. And what happens, the second round is always, remember, it is more powerful than the first round, it is more rapid than the first round, and it is much more effective than the first round. That is why it takes only 2 to 3 days, and the secondary response produces far more antibody or higher affinity and lasts longer. Memory B and T cells formed in the primary response drive the fast, stronger and secondary response. This is because of the memory B and T cells that the second response is so efficient, so effective and so fast. Okay. The principle is the foundation of vaccination. Right? Okay. Try to answer these two questions now. A key immunological difference between a primary and secondary antibody response is that the secondary response shows dash lag phase and dash antibody titer. Okay, the second one. Okay, so you're saying that it is B. Okay, good. The next one, try to answer the next one. Which of the following is an example of an antigen presenting cell that can also directly recognize antigen through its own antigen specific receptor? Its own antigen presenting antigen specific receptor. So try to answer. This is also very straightforward, very simple. Two is C. Yes, yes, very good. Yes, it is C. Right, because B cells, if you remember, they can directly bind to the antigen, right? So it does not need anything to be presenting this antigen. It can directly go and bind to the antigen. Okay, and the first one was shorter lag phase, that is very true, and higher antibody titer, that means a lot more antibody is produced per, let's say, ml. Okay, and a higher affinity of the antibodies. They are much more effective, right? Okay. Now, when immunity goes wrong, this is biology. 2 plus 2 is equal to 3.52, 4.5 in biology. And that is why, even though the system is so stringent, it is not foolproof, right? So, antigen type, normal response, and if the response is deficient or excessive, then what happens? Go through this table. So if there is an infectious agent, the normal response could be protective immunity, but there can be recurrent infections, right? If there is an allergen, normal response is allergy, but there can be absence of allergy, that is a benign outcome. Then there can be a grafted organ. So there is this, it's very difficult, like kidney transplant and all of these transplants that happen in our body, they just get rejected. So like it's very pathetic, but yeah, they do get rejected, right? That is because of a lot of different things that happen in our body. So we will study this in details in the upcoming classes. Okay, then there is something called self antigen. So what happens, there is self tolerance. You're not supposed to attack your own self antigens, your self proteins, but then that doesn't happen. There is autoimmunity, and autoimmune disorders are also very, very painful and very upsetting. Okay, but that is also that also happens, and we will study that in details too. Then tumor cells, tumors are supposed to be cleared, but cancer is, it just happens in such a way that the body cells are not able to identify that it is a tumor cell and it is not cleared, right? So antibiotics kill bacteria, but resistant strains are rising. Understanding and harnessing our own immune system is becoming even more important day by day. Right, now we have the last 2-3 slides. Okay, so virus, remember it is intracellular. For example, influenza, smallpox, chickenpox. So the primary effector mechanism would be NK cells and cytotoxic T lymphocytes. Intracellular bacteria, protozoa, for example, Mycobacterium tuberculosis, lepri, and then leishmania, etc. These will be acted upon by Th1 cells or IL1, ILC cells, right? Then you have extracellular bacteria and fungi, streptococcus, clostridium. I will have to make all of this in italics. Okay, so antibody plus your T helper cells and ILC cells, neutrophil recruitment. Then you have the small extracellular parasites, for example, trypanosoma, antibody, neutralization and opsonization. Now we talked about opsonization, but we did not talk about neutralization yet, right? So in neutralization, what happens is, let's say this is a toxin that is released by the pathogen. Inside the body, there will be an antibody or antitoxin that is going to come and coat this toxin completely and neutralize its effect. That is called neutralization. Okay, large parasites or worms, and they are helped with by the eosinophils and Th2 and ILC response. So different pathogens, lifestyle and demand different weapons. The immune system deploys them together, not one at a time. Okay. Now we have two more MCQs. Try to answer these, and then we'll come to the almost the end of today's class. One is B. Okay. Yeah. Okay. Great. Yes. The next one. Th1 cells help clear intracellular pathogen such as Mycobacterium tuberculosis primarily by directly lysing infected macrophages secreting IFNY, that is interferon gamma, which activates the macrophages to fuse phagosomes with lysosomes, producing IgE antibody against bacterium and recruiting eosinophil. Okay, so you're saying that it is B. Let's see. Yes, correct. So both answers are B. Very good. And we go to the next one. Next one is vaccination. Vaccination we have already talked about. So in vaccination, what happens? We are deliberately giving a primary exposure under controlled environment, safe conditions. This generates the memory B and T cells, keep them ready. On real or future exposure, the body mounts a fast, strong secondary type response, often preventing disease entirely. Best protection strategy we have is medicines can treat diseases and vaccination can prevent it, right? But research is ongoing for a lot of different disorders and diseases, for example HIV, malaria, a lot of parasitic diseases, etc., right? A lot of viral diseases also, Ebola virus and all of that. Now we have something called the clonal selection. So what's clonal selection? We said that now the B cell and T cell, they should not recognize our own cells as pathogen, as foreign, right? It should not recognize our own cells as foreign. And there is a very stringent checkpoint, a very stringent selection system that ensures that any B cell or T cell that recognizes the body's own cell as foreign is killed. Okay, either killed or rendered completely non-functional. So see what happens. In the developmental stage, each lymphocyte bear a single type of receptor with a unique specificity. Binding of a foreign antigen with that receptor with very high affinity activates the lymphocyte. Right? Effector cells derived from an activated lymphocyte bear receptors identical to the parent cell. Right? Self-reactive lymphocytes are deleted or inactivated early in development, absent from the mature repertoire. Repertoire means a group, huge group, right? So now this is the entire thing that is happening. So surviving mature lymphocytes are there and the different shapes are there showing that these are all antibodies having specificity for different antigens, right? So one foreign antigen binds and proliferates, and then what happens? That particular clone expands because that particular thing has attacked and we need to eliminate it first. Now this is shown in a very simple way, but this is the full procedure. It is huge and very elaborate, and we will be talking about this in the upcoming classes, of course. So this is how it happens. You can see. Okay. Right. So that is it. So clonal selection hypothesis is given here for your understanding, but I will not go into this today because we will talk about this in the upcoming classes. There is one MCQ that you can answer. Okay, so this is also regarding clonal selection. So try to answer. If you can answer this, very good. If not, no problem. We can talk about this in the next class. Okay. So in the clonal selection hypothesis, what is the fate of a lymphocyte clone whose receptor binds strongly to a self antigen during development? It is preferentially expanded to maintain tolerance. It differentiates directly into a memory cell. It is deleted or rendered functionally unresponsive or anergic. It switches its receptor specificity to a foreign antigen. Okay, try to answer this. And the second one is a deficiency in the mechanisms that maintain self-tolerance in developing lymphocytes. would most likely result in A is increased susceptibility to allergies only, improved graft acceptance, autoimmunity, and complete loss of innate immunity. 2 is C. Okay. What about 1? Try answering. This is not difficult. We have talked about this. Try answering this. It is doable. It is doable. Chalo, try. No one is trying. Why? There is nothing to lose. Come on, try. One is C. Yes. Okay, very good. Yes. Always try. Trying is very important in life. So C. Yes. C and C. Both are C. Okay. All right. So that's it. And neutralization. So neutralization, I've already told you what neutralization is. It is given in details here. Okay. So I will be sharing the slides with you anyway. So you can go through it whenever you get time. Now we have a few more questions. Try answer. Okay. No. Okay, there are questions. Okay, so we will actually go through this then. So neutralization and opsonization. So neutralization is an antibody effector function in which the antibody binds directly to the pathogen or toxin in a way that physically blocks its ability to interact with the host cells, okay, without requiring any additional immune machinery. For example, antibodies can bind to a virus's surface protein and prevent it from attaching to or entering the host cell, or bind to a bacteria's toxin, for example tetanus toxin or diphtheria toxin, and prevent it from binding to the cellular receptor. So how does the toxin work? So let's say this is the bacteria which release some toxin, and let's say this is a cell, this thing is my cell, this is the receptor on the cell. The toxin comes and binds to the receptor. As soon as it binds to the receptor is going to take it inside, and what is going to happen? It is going to affect the cell in some way, right? So for example, tetanus toxin, this one, it gives you this lock jaws. The entire muscles in your body, they become very, very stiff and they cramp up, and it is pathetic. So people die from this bending of their back, and it is terrible, right? So what happens is, um, this particular in this method, antibodies go and coat the toxin completely. So now the toxin is not able to bind to the receptor anymore, right? Now what is happening? See, rendering it harmless even though the toxin molecule itself isn't destroyed. Because neutralization works purely through steric blocking, physically occupying the binding site of the pathogen or toxin needs, it does not require complement or phagocytosis. The antibody alone is sufficient to stop the infection or toxic process, and this is the primary mechanism behind many vaccines. Tetanus toxoid, you know already, right? Where neutralizing antibodies generated by prior exposure or vaccination prevent the pathogen from ever establishing infection. And that is why tetanus toxoid you're supposed to take every few years or so, every few months, every few years. Now, opsonization, this we know already. So opsonization is a process where the pathogen is coated. Okay, brilliant. So the pathogen is basically coated. Now we have discussed opsonization. So you can try to answer this question. Okay, try to answer this question. Try to answer this question. I'll be back. Yes. Okay. Anyone answered? 2 is B. Acha. Okay. Answer will be B. All right, great. So antibody mediated opsonization enhances pathogen clearance mainly by coating the pathogen so that the phagocyte recognizes it via Fc receptor and engulfs it more easily. Okay. The next one is the mechanism by which the antibody prevents itself from bacterial toxin from engaging it with its host cell is again neutralization. So both are B. Fantastic. Right. So yes, that is very correct. Both are B. Now, here is the complement system. Now, complement system we will not discuss about today because we will discuss about this in the upcoming classes in much details. Okay, so I'm skipping this portion. There are 2 slides. You can understand how huge this is. Okay, so we will be talking about this later on. And here are your references. Okay, so please refer to this book. And the first link is the web page of this online copy of this book basically, so you can refer to that. And then there are a few MCQs and all given, so all of these different links you can refer to. Thank you. That's all. If you have any questions you can ask me now, and I'm giving you my mail ID so you can actually mail me if you want. Okay, so any questions that come to your mind during the week, please mail me at it. And right now if you have any questions you can ask me anything that you want to share, anything that you want me to improve, be it my slides, be it my teaching, anything. Okay, so you can tell me. Okay, that's it. This is my mail ID. Okay, you can send me mails and in the next class we can discuss. I can also reply you via mail. Okay, it will be confusing in the beginning, but read the book and you'll understand things very well. Okay, thank you. If nobody has any questions we can end the class today. Okay, so let's end the class. If you guys don't have any questions, we can end the class. Otherwise, you can ask me in the chat box, okay? Or you can even unmute yourself and you can ask, no problem. And I've given you my mail ID, so thank you for your time. Thank you for your attention. Thank you for attending the class and being so attentive, answering all the questions. And that also correctly. Very good. Okay, so that's it. We'll be ending the class then since no one has any questions today. Thank you so much. Bye-bye. Have a good day.